My name is Amy Ryan. I'm an assistant professor of medicine and stem cell biology and regenerative medicine here at USC. I come to USC with a background in a master's and PhD in pharmacology from the University of Bath in the UK, where I focus mainly on biophysical approaches for analysis of the pulmonary vasculature. I continue to do my postdoctoral research by moving over to UCSD, where I investigated pulmonary vasculature, and then a second postdoctoral position at the Salk Institute of Biological Studies in San Diego, where I moved more into looking at stem cells and pluripotent stem cell models of airway disease. Since then, I've been recruited to USC, and I've been an assistant professor here for 5 years now. I'm a member of the Hastings Center of Pulmonary Research here at USC, and also outside of USC, I'm a member of the Cystic Fibrosis Foundation Epithelial Stem Cell Consortium, the USC's COVID-19 task force in the past year, and I'm also chair of the Stem Cell Therapy, Bioengineering and Lung Biology and Diseases Conference, which is held every 2 years. In addition to that, I'm also on the editorial board of Cell Stem Cell and the American Journal of Cell Physiology. So my lab, our research focus is really on the development and utilization of pluripotent stem cell models to study human lung diseases, which is really addressing a critical lack of model systems that closely reflect the disease pathogenesis as it occurs in humans. Our primary research focus is on rare lung diseases, where it's very difficult to obtain human samples, either from the patients or from end-stage disease. We're particularly focused on looking at cystic fibrosis and another airway disease called primary ciliary dyskinesia, which affects the ciliogenesis and mucociliary clearance in your airways. Another focus of the lab is developing multi-scale models to investigate cellular interactions, particularly immunoregulation and physiological biomechanics of the airways, regulating the repair and regeneration of the airways after injury. A project that I'd like to highlight to you is one that we've been working on since my postdoctoral years at the Salk Institute, which is really perfecting this generation of a functional basal airway stem cell from the pluripotent stem cells. We're really limited right now, as the state of the art for doing this is use of primary airway basal cells that are isolated from explant lungs. And these are obviously limited in availability. The quality of the isolations can vary, which changes reproducibility, and also representation of different disease-causing mutations, particularly rare mutations causing primary ciliary dyskinesia and cystic fibrosis are hard to obtain. So our approach is to generate pluripotent stem cell-derived basal cells that can be gene-edited and represent a spectrum of rare lung disease. And that could also be then adapted and readily used in high-throughput screening approaches. The limitation that we're trying to overcome right now is that current protocols are still relatively inefficient, and they tend to produce basal cells, which are the main stem cell of the airways, that have variable differentiation and functional capacity. So to date on this project, we've modified our protocol and successfully defined a new protocol for generating airway basal cells that have the core phenotype of airway basal cells. But unfortunately, still the functional differentiation is inconsistent, so we've been investigating why. We believe that this cellular state is driven, at least in part, by the biomechanical properties of the extracellular matrix that the cells have been grown on in 2D, as we see very distinct differences when we culture the cells in 3-dimensional spheroids versus in regular 2D cultures. So overall, right now, our data is indicating that induced iPSC-derived basal cells may be more sensitive to stress signals, particularly in regular 2D culture than human primary basal cells. And one of the things we're investigating here is whether this is epigenetically regulated, whether the iPSC-derived basal cells are somewhat epigenetically naive because they haven't been exposed to those cellular interactions they would be inside the human lung. So as for current collaborations, we have a number of very productive collaborations, both within USC and across the United States. One of those is with Kalpaj Parekh, and we're working with him at the University of Iowa, looking at ferret models for cellular engraftment of both primary and iPSC-derived ferret basal stem cells. We work with Chris Kintner at the Salk Institute, looking at mechanisms of multiciliogenesis. And in the same vein, we're working with Eva Cancel in the Viterbi School of Engineering at USC, where we're developing techniques for quantitative analysis of functional mucociliary clearance. Recently, we've established a collaboration with Sumit Chanda, where we're looking at high-throughput screening for therapeutic options for COVID-19. And then obviously we have a very rich collaboration with the members of the Cystic Fibrosis Stem Cell Consortium. Currently, my funding is primarily from the NIH, with R01's funding work on primary ciliary dyskinesia and ciliogenesis, and the Cystic Fibrosis Foundation, the consortium funding there for our work on cystic fibrosis stem cells and looking at niche regulation of stem cells. And then over the past year, we've acquired some pilot funding to look at COVID-19 from the Rainwater Foundation and the Keck Foundation. But always we're looking for productive collaborations that would lead to advancing therapeutic options, in particular for these rare lung diseases that don't have a lot of therapeutic options currently. Thank you very much for your time.